the last step in the process of protein synthesis is translation. And I'm going to divide translation into 2 sessions. So in the first session, I'm going to discuss about the meaning of codon or triplet and how can we match the codon into its correct amino acid and what are the start codon and the end codons. So mainly when we talk about translation, we speak about the formation of a protein. And this is the last stage in protein synthesis. Recall that we started with DNA replication, then transcription, and now translation. So what was the end product of transcription? If you remember that when transcription ends, we had a messenger RNA molecule that was processed and ready to exit the nucleus into the ribosome. So let's take, for example, this messenger RNA molecule. As can you see, it's single strand and it has U instead of T. The question is, how is this messenger RNA molecule scanned in the ribosome? So after it goes to the ribosome, how is it going to scan? And how is the information taken from this messenger RNA in order to make a protein? Before we explain about all of this, what creates a protein? What are the monomers of protein? The monomers of proteins are amino acids. And in our body there are 20 types of amino acids. All amino acids have in common 2 groups. We call them amino group and carboxyl group. Amino group and carboxyl group, as can you see here marked in blue and in green. So what is the difference among those amino acids? There is an R group, or there is a chain connected to the carbon in the middle. This is different from one amino acid to another. Some of those R groups are basic, some are acidic, some are polar, some are non-polar. So there are different properties of amino acids. What you have to know so far that getting those amino acids and linking them together by a peptide bond will create a protein. Okay? Now, since we have 20 different types, those 20 different types are used in a certain sequence, certain order, in order to make a unique protein. So what is the function of the messenger RNA in this process? The messenger RNA is going to decide which amino acid is going to be the first, which amino acid is going to be the second, and so on, until we produce what we call a polypeptide. What is a polypeptide? A polypeptide is a protein primary structure. It's the basic structure of the protein. I'm calling it now here protein, but later on we'll study that there are more modifications and changes that happen to the polypeptide to become a protein, which is mainly folding and taking a 3-dimensional structure. So let's go back to our main question. How is this messenger RNA molecule scanned in the ribosome? So after this messenger RNA goes into the ribosome here, so after the messenger RNA goes into the ribosome, amino acids must be brought together in order to make the protein. How does this happen? In order to understand it, we need to define a new term called codon or triplet. Triplet means 3. So to start, we have to look at this messenger RNA as triplets. And here what I did is I divided this messenger RNA into triplets, 3 and 3 and 3 and 3. For example here, AUG, CUA, GGC, UCU. It's the same as this one, but all what I did is I divided them into triplets. What do we call those triplets? We call them codes or codons. Now, every codon is going to code for a single amino acid. Every codon will code for a single amino acid. And there is a chart that you'll always have it as a given. You don't have to memorize it. So this is a universal language, universal chart that shows which amino acid is coded by which triplet. For example, AUG. Look at AUG. AUG, there is the amino acid MET. What is MET? It's called methionine. This is MET here. MET. For example, LEU is leucine, ASN, asparagine. So, the second codon or triplet is CUA. C is the first letter, so this is the first letter C. Then U is the second letter, this is U, so I'm looking at this square, and A is the third letter, CUA. CUA codes for leucine. And as you notice here, there are 1, 2, 3, 4, 5, 6 codons or triplets that code for the same amino acid, which is leucine. There are as well 1, 2, 3, 4 that code for proline. There are 4 that code for SER, serine, etc. So as a total, we have 20 amino acids, and we have here 64 codons. 4 times 4 times 4. So we have 64 codons, all the possibilities of having triplets. Those 64 codons code for only 20 amino acids because we have multiple codons that code for the same amino acid. So as in total we have 20 amino acids. So this is to answer the question, what is a codon and how can we match the codon into the correct amino acid? We do that by simply referring to the codon chart. And again, this chart always is given. You don't have to memorize it. So whenever you have a question, an assessment, you'll be having this chart in the assessment as well. So if you have the messenger RNA sequence, you are going to divide it into triplets, 1, 2, 3, etc. How many triplets? Then you are going to match every triplet into the chart to look for the specific amino acid. So for example here, AUG, I'm writing methionine, this is the amino acid. CUA, it's leucine, this is the amino acid. GGC, it's glycine, this is the amino acid. UCU is serine, and so on. Now, the last question we asked here, what are the start codon and end codons? Among all those codons, you don't have to memorize any of them, but there are 4 codons that are remarkable. 3 of them are called stop codons. It means they don't code for any amino acid. And there is only one, and here it is colored in red, AUG. This is the start codon. What do I mean by start codon? All proteins start with AUG or with methionine. So it's always going to be the first amino acid in any protein. Why? Because it marks the start of translation. So whatever is before or after, always it's going to start with AUG. Now, what about the rest? Stop codons. What are those? Stop codons. Whenever there is a stop codon, translation is going to stop. It means after that, no more amino acids are added to the sequence of amino acids. So look at that. We started here, translation. This is translation. It means translating the messenger RNA into amino acid based on the table. After I reach UGA, if you look here, UGA is called stop codon. So I stopped here. After that, I didn't continue translation. This will mark the end of translation. And this molecule of 1, 2, 3, 4, 5, 6 amino acids is now called the polypeptide, or this is the protein. Okay, so in brief, how proteins are made? Simply in the ribosome, by scanning the messenger RNA and matching amino acids based on the triplets. The first amino acid to start with is always methionine, that is coded by AUG, and the amino acid sequence will stop when there is a stop codon, and there are 3 stop codons in the chart: UAA, UAG, and UGA. Whenever those are reached, the amino acid fragment is going to stop here. So this is regarding what is meant by codon and how to match the codon with its correct amino acid and what are the start and the end codons.